This is a production of Cornell University.
Thank you so much for joining us today. My name is Tony Bracco, and I work alongside Dr. Zachary Stansel, who's the USDA Hemp Germplasm Curator. And I wanted to tell you a little bit about our work and why we wanted to host this hemp webinar series. And so my team and I run the Hemp Germplasm Repository, which is similar to a library, but for seeds. And the first goal of our work is to help protect the genetic diversity of hemp. And our second goal is to make a lot of different types of hemp available for science. And so these seeds can be requested by plant scientists, plant breeders, and educators. And we believe that these seeds can be useful to answer all sorts of questions. And we hope that this work allows universities, companies, farmers, and everyone else to benefit from the 1,000% potential uses of hemp. During this work, we've met so many interesting and dynamic people across the world of hemp, and we wanted to present them to you. So we teamed up with our friends at Cornell University to present them to you today. So I want to give a huge thanks to George Sack, Gemma Osborne, and Kim Paul. But most of all, I'm thrilled to present our speaker today, Dr. Daniela Vergara. Dr. Daniela Vergara is an evolutionary biologist, data analyst, educator, scientific writer, and public speaker. In addition to her multiple publications, she founded and directs a nonprofit organization, the Agricultural Genomics Foundation, which aims to make hemp and cannabinoid science available to a broad public. Dr. Vergara has been part of scientific teams at private companies, including Steep Hill and a biotech company called Front Range Biosciences. Through AGF, Dr. Vergara educates the public about science, data analysis, statistics, evolutionary biology, and genomics. Dr. Vergara is also an international consultant and policy advisor and has advised companies in the US and Latin America. Additionally, she has worked as an expert witness for multiple legal cases in the US and abroad. Dr. Vergara is a scientific writer in both Spanish and English and has translated her scientific work in Spanish for a broad public in Hispanic countries. And so before we begin, I ask that as you think of questions for Dr. Vergara throughout the presentation, please put them in the Q&A tab, and anything written in the chat tab will not be looked at in the Q&A at the end. So please put your questions in throughout the presentation. And without further ado, take us away, Dr. Vergara.
Thank you very, very much, Tony, for the very nice presentation, and thank you for the organizers. So thank you very much. I am Daniela Vergara. I'm the emerging crops specialist at Cornell. This is my email, my website, my Twitter, if you want to contact me. So as Tony was saying, I was part of the University of Colorado Boulder. I still am a lecturer there. I teach a class for the broad public through continuing education if anyone wants to take it. I am the co-founder of the Cannabis Genomic Research Initiative, which is our group at CU Boulder that aims to study the cannabis genome. And I co-founded the Agricultural Genomics Foundation, which is a nonprofit organization that I currently direct that aims on bringing science, cannabis science to a broad public. And right now I'm part of the Cornell Cooperative extension. So if you looked at the schedule today for 20, lucky me, I was scheduled to give a talk on hemp diversity and genetics. And due to my background, the first thing that I thought was to talk about the cannabis genotype. However, today is the birthday of this paper that got published last year. So it's 1 year since I published this paper. So I wanted to talk about this paper that deals with the phenotype. But, you know, the talk was on hemp diversity and genetics, so I needed to talk about the genotype. But in my opinion, one of the reasons why we study the genome, which is the whole conglomerate of DNA, all of the collections of letters in a genome, right? Each of us has a genome. Cannabis also has a genome. Each plant has a genome. So I wanted to talk about the genotype. And one of the reasons we study the genome is to relate it to the phenotype. So today I'm going to be talking about the cannabis genotype and phenotype. And I want to let you know that my talk is going to be a little graph heavy, but I'm going to try my best to explain each of the graphs. And I want to start with acknowledging all of my collaborators from all over the place, Leafly, the Agricultural Genomics Foundation, CSU Pueblo, CU Boulder, of course, which is the place where I produced all of the data that I'm going to show you today. And of course, Front Range Biosciences, where I worked for 2 years. And my talk is going to have 3 different parts. I'm going to talk about the cannabis genome, the cannabinoid genes, the terpene genes, and how each of these relates to the phenotype. And I'm going to start with a take-home message. When in the industry they tell you about sativa and indica, be skeptical. So I'm going to start with the cannabis genome. So cannabis is an angiosperm plant. So it's a plant that produces flowers. It is part of the cannabis of the family Cannabiaceae. Cannabis sativa L was described by Linnaeus in 1753. Cannabis is dioecious, so males and females are in different plants, kind of like us, right? We have males and females in different individuals. Males have an X and a Y chromosome, like us, and females an X and X chromosome, 2 X chromosomes. There's also monoecious individuals. So monoecious individuals produce both males and female flowers, and they have undifferentiated chromosomes, which means that they're not like the X and the Y, but the chromosomes themselves are not different from each other. And apparently, in these monoecious individuals, part of the chromosome is similar to the female chromosome, at least a part of the chromosome is. And cannabis is diploid, right? So it inherits, like us, 1 set of chromosomes from mom and 1 from dad, right? So part of the chromosomes come from dad, half of the chromosomes from dad, and half from mom. The genome size in cannabis, females have approximately 818 megabase pairs, so 818 million letters in the genome. The males have 843 million letters, so the genome of the males is larger than that of the females because the Y chromosome is larger. And in fact, the Y chromosome in cannabis is the largest of all of the genome. And again, it has 10 chromosomes, 9 of them autosomes, so 9 of them are not related to sex chromosomes, and then it has an X and a Y. So when we sequence a genome, when we take all of the letters of the genome, of the DNA, and we transcribe them, we have to break them up into little pieces. And then we have to put them back together bioinformatically, kind of like putting together a puzzle, right? So ideally, when we have something that it's called a genome assembly, we would have 9 autosomes, so 9 chromosomes, 9 pieces that are not related to the sex chromosomes, and then the sex chromosome. So in total, we would like 11 total pieces with the X and the Y, right? 9 autosomes plus X and Y, with the 843 million base pairs. And indeed, right now, that is where we are, more or less. Thanks to Grasa and collaborators from 2018 in their preprint, and then later in 2021, we definitely have 9 autosomes. So we have 9 chunks of DNA, and we have 1 sex chromosome. We are still lacking the Y chromosome, but we still have 200 pieces that we don't know where they go, right? So we're very close of having a very good assembly, and Grasa's assembly is a very good assembly. And this is how it looks like. We have chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, and the X chromosome, right? We're still lacking the Y chromosome. And we not only have 1 assembly, right? We have multiple assemblies. I think that right now we have between 13 to 15 assemblies that are publicly available. Here in this slide, I'm showing 9 of them, right? So 9 individuals, right? 9 individuals. So we have an individual purple kush, pinola, Jamaican lion, pineapple, banana, baba kush, CS10, which is the one that Grasa assembled, the one that I was just talking about, Canatonic, Carmagnola, USO31 from CSU Pueblo. I'm not sure if they're already publicly available, but I'm sure they will be soon. And this supposedly wild assembly that is also in very good state. So I think that these two, CSN and the wild one, we don't really know if there's wild cannabis, that's why it's wild, but that's what they say. These two are the ones that are in the best shape. So here I am showing 9 of the 13 or 15 assemblies that we have available. So when we look at the relationship between cannabis varieties, cannabis strains, we see something like this. This is called a split tree graph. Each of these lines is a strain. The closer you are in this graph, the more of the genome you share. So if my son and I were in this graph, we would be very close to each other because we share a lot of our genome. So each of these lines is a strain. I'm showing here the relationship between approximately 300 different varieties, 300 different strains. This thread-like structure in the middle is the part of the genome that everyone shares. So it's the ancestral part of the genome. And depending on how you divide this graph, you more or less see these 3 groups. This is how we divided this graph. And we can see here the broadleaf marijuana types, the narrow-leaf marijuana types, and the hemps. The broadleaves and the narrow-leaves, the broadleaves in purple, the narrow-leaves in blue, these two are used in medical and recreational settings, while the hemps are used in industrial settings. And I want to clarify something. These hemps for industrial settings, this group that we see here, are the ones that are used for fiber or for oil. So these guys over here are not necessarily the ones that have, I mean, usually they have less than 0.3% THC, but the government coined this same hemp for anything that has less than 0.3% THC. And there are some individuals that are not necessarily hemp, that are actually low THC marijuana, individuals that even though they have less than 0.3% THC, they fall in these categories over here in this graph when we look at the genome. Now, when we look at these 9 assemblies that I just showed you, these assemblies also fall into some of these groupings. So we have, for example, purple kush and pineapple banana baba kush that are part of the broadleaves, Finola, Carmagnola, and USO31 that are part of the hemps, Jamaican lion and Canatonic that are part of the narrowleaves. And then we have these 2, CSN and the wild one, that are hybrids. CSN indeed is a hybrid between a hemp and a marijuana type. And Finola and purple kush, I want to say these were the first 2 assemblies that we had available. They were published in 2011 by a Canadian group led by Jonathan Page. And then in 2019, they improved these assemblies as well. When we look at the variation in the nuclear genome, and these are preliminary results, we have in this graph, we have our 3 groups, the broadleaves, the hemps, the narrowleaves. And we have in the y-axis, which is our vertical axis, we have diversity. When we look at the diversity in cannabis, we more or less see that it has approximately 2 to 3% nucleotide diversity. This is a measurement of genomic diversity. And this doesn't mean much, but when we compare it to other species like bonobos, chimpanzees, humans, domesticated corn, we can see that all of these species have less than 1% diversity. So cannabis has approximately 2 to 3% more diversity than any of these species. And we can see that as well in the phenotype. These are leaves from different strains, from different varieties. And we can see that the color varies, the number of leaflets varies, the size varies, and the leaves, all of these leaves are very different from each other, but the plants themselves are also very different. The height, the colors, the branching, and of course, the variety of compounds that cannabis produces. Cannabinoids, depending on how you count them, can be up around the hundreds. Also terpenes and flavonoids. There's a bunch of different compounds. And of course, the plant can be used for multiple purposes. The fiber can be used for rope, for paper. The oil can be used in personal products. Hemp seeds are edible. It can be used for biofuel, for clothing, and of course, for medicine. In the industry, we're used to see these two names, indica and sativa, and then you have the indicas are supposed to have dense buds, and the plants are short, and the leaves are bushy, and then the sativas are tall, and the leaves are thin. But when we look at leaf shape, I'm going to be showing this type of graphs several times throughout the talks. These are called principal component analysis graphs, and these are a dimensionality reduction graph. So we're basically taking a bunch of variation and showing it in 2 dimensions. Principal component 1 in the horizontal axis, the x-axis, and principal component 2 in the vertical, the y-axis. And in this case, we are showing the variation in leaf shape. Okay, so we have different points in these leaves. These points are called landmarks. We have different dots in these leaves. And if you are in this side of the graph, you have a squat leaf and your leaflets are pointing out. If you're in this side of the graph, you have more of an elongated leaf and your leaflets are pointing in. And between these 2 principal components of all of the variation, right, between these 2, we're explaining more than 80% of the variation, which is pretty high. Females here are the circles, males are the squares. And if we had differences between sexes in leaf shape, we would expect, for example, that the males are in this side of the graph and they have a squat leaf, and the females are in this other side of the graph and they have an elongated leaf. Or perhaps the other way around, where the females are in this side of the graph and have a squat leaf, and the males in this other side of the graph with an elongated leaf. But what we see is that there's no correlation whatsoever between females and males and the leaf shape. So you can be a male and have a squat leaf, or you can be a female and have a squat leaf that has no relationship whatsoever between sex and leaf shape. And in fact, we see that leaf shape has no relationship with height, no relationship with branches, and no relationship with a bunch of other traits that we measure: stalk diameter, number of nodes, petiole length, bud count. It has no relationship. This is all data from birthday paper. And that is probably due to recombination. Recombination is this wonderful process that sexual individuals undergo. We have, again, a chromosome from dad and a chromosome from mom, and then a chunk of the chromosome swaps. So we get a chunk of the chromosome from mom in dad's chromosome, and etc., right? Recombination kind of looks like this. It's not just one little chunk, but there's multiple places where these chromosomes recombine. And thanks to recombination, we have a bunch of variation. And thanks to recombination, we differ from our siblings, right? Is one of the ways in where we differ from our siblings. And maybe we share, I don't know, hair color or the shape of our eyes, but we are different from our siblings. And recombination is one of the reasons. And thanks to recombination, we have, red hair with blue eyes, brown hair with blue eyes, blonde hair with blue eyes, and so on. It's a great way to shuffle genes around and to create novel combinations. So when they tell you that indicas are tall and have this particular leaf shape, and sativas are short, whatever it is, be skeptical, because all of these traits can be shuffled around. Especially all of these compounds, because cannabis, again, produces a bunch of different compounds. Which leads me to the second part of my talk about cannabinoid genes. But to conclude this first part, cannabis has a lot of genomic variation, a lot of phenotypic variation. A bunch of these phenotypic traits are not correlated to each other, can be shuffled around and broken by recombination. Perhaps many different combinations are possible through breeding, right? Many different combinations that we do not see yet. And the genome assemblies still need a lot of work. We have very good assemblies, but we still do not have a good assembly of the Y chromosome or of the monoecious chromosomes. So now I'm going to start talking about the cannabinoid genes. Okay, so cannabinoid genes. So this graph is a very important graph. I've shown it in multiple talks. I want to clarify something. This is just the last snippet of a huge biochemical pathway, right? This is just the last step of this huge biochemical pathway. When we talk about cannabinoid genes, we're talking about the genes that code for these enzymes, these three syntheses, right? I'm going to talk about three cannabinoids of the multiple cannabinoids that cannabis produces, and those are THCA, CBDA, and Cbca. So these enzymes are proteins that perform a job. They do something. In this case, these syntheses, these enzymes take Cbga, so cannabigerolic acid, and convert it into either THCA, CBDA, or Cbca. Now, from this pink side up, all of this happens inside of the plant. And when we heat these compounds up, when we heat them up, they turn into THC, CBD, and Cbc, but this is outside of the plant, right? So through heat, that's why baking, vaping, smoking, we heat them up and they turn into the neutral forms. So the neutral forms, right, tetrahydrocannabinol or cannabidiol, they lose this COOH, right? They go into the acidic form, into the neutral form. And again, this happens outside of the plant. These neutral forms are the ones that interact mostly with our endocannabinoid system. However, there is some evidence that the acidic forms also interact with the endocannabinoid system. But in any case, we have that these three enzymes use the same precursor molecule. They use Cbga. So under that definition, these three enzymes could be considered promiscuous enzymes because they are using the same precursor molecule. Now, when I was an undergrad, I was taught that enzymes are very specific. But this is not the case of these enzymes, at least. Apparently, it's not the case in general, but at least not of these enzymes. We know that these enzymes in vitro, and this is thanks to a paper, Serpel et al. 2018, they show that these enzymes can produce up to 8 different compounds, including each other's compounds. So in other words, THCA synthase can produce a little bit of CBDA, CBDA synthase, a little bit of Cbca, Cbca, THCA, and so on. So these enzymes can be promiscuous and can be sloppy. Okay? So I want to define something. A gene is in a location in our chromosomes, right? So this is the same location for mom's chromosome and for dad's chromosome. And so it's a locus, right? So when we're talking about a locus, it's a specific location. And an allele is a form of a gene. And in this case, mom or dad could have the same allele, the same form of the gene, or they could differ. And at the beginning, we thought that if dad had both THCA and CBDA, so if dad was heterozygous and mom was also heterozygous, then the offspring, 1/4 would have THCA, 2/4 would produce both compounds, and then 1/4 would produce CBDA, right? So we thought that this was a one locus, two allele type of thing. It was the same gene that had 2 different forms, THCA and CBDA. And that's where we have this type 1, type 2, type 3, right? Type 1, high THCA, type 2, both THCA and CBDA, and then type 3, CBDA. And it wasn't until 2015 with a really good paper by George Weinblain, where they found one individual, they made a cross between a marijuana type and a hemp type, which is RCS10 assembly. So they made this cross, and there was one individual that recombined. They had less than 100 individuals. I think it was around the 60s or the 70s. And there was one individual that got the THCA and the CBDA loci. One individual recombined. And thanks to that one individual, we found that these were 2 different genes that were in close proximity, approximately 1 centimorgan apart, so 1 recombination event apart. So thanks to this recombination event, we now found that indeed these were 2 different genes that were in very close proximity, and that it was really hard to tear them apart because they were in close proximity. Again, when we used to play chain when we were kids, or maybe you still play chain right now, I'm not sure, but when we used to do that, it is harder to break people apart when they're close together. It is easier to break them apart when they're far apart. That's the same case as CBDA and THCA. They were very close together, and therefore it was very hard to tear them apart. So thanks to this one individual, this recombinant, Grassa used the same population with his paper. This is a paper that I was just talking about. George Weinblain is also part of this publication. And this is a very busy figure, but what I want to take, what I want you to take from this figure is that these genes indeed are very similar to each other. This shows the relationship. This part of the figure shows the relationship between the individuals. They're very similar to each other, and they are in very close proximity in the genome. All of these cannabinoid genes are in very close proximity in the genome. And this is what we see in their map. We see that all of the cannabinoid genes are in this chromosome. So at least the genes with major effects, the genes that have the most effects in cannabinoid production are on this chromosome. But there are multiple genes all over the genome that are related to cannabinoid production. Then in 2019, we had a paper where we discussed that all of the individuals have, all of the cannabis varieties differ in the number of cannabinoid genes. All of the different strains have different number of genes, and that this variation in copy number in these genes could have associations to cannabinoid production. And indeed, when we look at the different assemblies, we can see that all of the assemblies differ in the number of genes, right? And we have, for example, this wild one that has 8 copies of cannabinoid genes, and Jamaican lion that apparently has up to 32. I also recommend this other paper that talks about cannabinoid genes, Van Velzen and Schranz, published in 2021, that talks about the evolution and the origin of these genes. This is another must-read paper. So now we know that this THCA, CBDA being one gene, being one locus, 2 alleles is not the case. So when we look at the genes, the cannabinoid genes from all of these different assemblies, and we look at the relationship, again, I want to make it clear that these are 9 individuals. These are 9 individuals that I'm showing in this slide, that these 9 individuals differ in the number of cannabinoid genes. But when we look at the relationship of all of the genes from these 9 individuals, this is what we see. And I'm not going to go in depth into this graph, but what I want you to take from this graph is the following. There's a bunch of genes related to CBCA production, despite CBCA being produced in very low quantities. There are some genes related to THCA production, and most of the individuals that have THCA are marijuana type cannabis. There's a bunch of genes that we don't know what they're doing. We have no idea. And every single one of the assemblies has a gene that falls into this category, in this unknown category. And there's some genes related to CBDA production. Some hemp have CBDA, some marijuana types have CBDA. So again, something that really caught my attention was, for example, Cannatonic. Again, this is one individual of Cannatonic, right? So I'm drawing a bunch of conclusions based on one individual. However, Cannatonic in the industry, it's sold as a 1-to-1 strain. So it's supposed to have one CBD and THC in equal proportions. However, Cannatonic has a truncated CBDA gene, so an incomplete CBDA gene and an incomplete THCA gene. So it makes us wonder, right, if that one individual of Cannatonic, if there's other genes that may be coming together to form this cannabinoid phenotype, right? So we have then 2 genes or multiple genes that come together to form the phenotype. And in biology, we call that epistasis, where genes interact to form a phenotype. So perhaps all of these genes are interacting to form the phenotype, right? So what does the phenotype tells us? So this is a paper that is in press right now with my collaborators from Leafly. Here in the vertical axis, the y-axis, we have cannabinoid concentration. And here in the x-axis, we have the different cannabinoids. And so this figure is a little biased because this is the cannabis found in dispensaries. So of course, the cannabis found in dispensaries have high THC. And we are not taking into account cannabis from hemp, right? We do not have hemp for fiber or for oil. It's not represented in dispensaries. But the cannabis from dispensaries, we have a lot of THC. The median is between 15 to 20, maybe 18%. Some individuals produce a lot of CBD. There's some CBG, but really most of the cannabinoids are negligible, right? Again, it's interesting that CBC is produced in very low quantities, despite there being a bunch of genes related to CBC production. And if we look at the relationship between THC concentration in the x-axis, the horizontal axis, and CBD in the y-axis, the vertical axis, we see that there's some individuals that produce a lot of THC and very little CBD, some individuals that produce a lot of CBD and little THC, and some individuals in between. Now, if we look at leaf shape and cannabinoids, right? So again, males are the squares, females are the circles. And if we look at leaf shape and cannabinoids, perhaps we would be expecting two groups, right? That maybe these individuals over here have a particular chemotype and have a squat leaf. These individuals over here have another chemotype and an elongated leaf, or maybe even 4 different groups with 4 different chemotypes and 4 different leaf shapes. But that's not what we see. We see that leaf shape has no correlation whatsoever with cannabinoid content. So you can have high THC and have an elongated leaf, or high THC and a squat leaf that has no relationship whatsoever. So again, when they give you, when they tell you that indicas are one way and broadleaves and high cannabinoids and sativas are another way, be skeptical. However, we do see between cannabinoids here in the x-axis, in the horizontal axis, and terpenes, we do see that there is a correlation. So the more cannabinoids you produce, the more terpenes you produce. Now, this correlation is not very strong, but it is significant, which makes us wonder whether there are some genes that are related to both cannabinoid and terpene production. And I can think about some, for example, trichome density or trichome number, right? We know that in the trichomes is where we have the production of all of these compounds. Most of the trichomes are abundant in female flowers. So perhaps if you have a lot of trichomes, or if you have big trichomes, you're gonna produce both a lot of cannabinoids and a lot of terpenes. And Zagar et al. In 2019, they do suggest that there is an interaction between genes, that there is a network between genes that are acting together to produce the cannabinoid and the terpene phenotype. And this is another must-read paper, in my opinion. Which leads me to the last part of my talk about terpene genes. But to conclude this part, there are many genes related to cannabinoid production. All of these genes are interacting to produce the phenotype. That's still a question. Individuals differ in the number of cannabinoid genes, so there is copy number variation associated to cannabinoid production. And cannabinoids and other phenotypes are not related to each other. So terpene genes. And I want to start with this graph, again, showing the relationship between terpene synthase genes, TPS. And this is by my colleagues at Front Range Biosciences. So something that I want you to see from this graph is that it appears that there's many genes related, for example, to the production of beta-myrcene, or to limonene and beta-ocimene, right? And if we look at this other paper by Booth et al., again, 2019, I think that 2019 was the year of the terpene. So we see that in the different chromosomes in cannabis, there are multiple genes related to terpene and cannabinoid production. So all of these genes are scattered throughout the genome. Again, supporting that idea from Zager et al. That there are multiple genes acting in unison to produce this cannabinoid and this terpene phenotype. So I talked to you about epistasis, where it's genes interacting, but perhaps we also have pleiotropy, where we have one gene that is affecting two different phenotypes, right? So the expression of multiple traits by one gene. So perhaps what's going on with the terpenes is that, for example, we have TPS5 that is related to the production of beta-myrcene, but we also have TPS23 and TPS12, and perhaps we also have the TPS5 is related to the production of limonene and beta-pinene, and then TPS12 is also related to the production of osamine, and so on, which is what Zager et al. Are proposing. So maybe we have, between cannabinoids and terpenes, we have pleiotropy and epistasis in place, genes interacting to produce this phenotype. So now, what does the phenotype tell us? And again, this is a graph from our Leafly paper that is only analyzing the cannabis from dispensaries. So it is a biased sample, but the cannabis from dispensaries, this is what we see in the y-axis, the vertical axis. We have total terpene concentration, and here we have all of the different terpenes. And we see that the cannabis from dispensaries is high in beta-myrcene, beta-caryophyllene, and limonene. So if you go today at a dispensary, it's likely that you can buy something that has high beta-myrcene, beta-caryophyllene, or limonene. Now, I'm going to show another principal component analysis graph. Here, again, this is a dimensionality reduction, and each of the dots is an individual. And PC1, principal component 1, explains the most variation, then PC2 explains the next more variation. And between these two, in this particular graph, between these two, we're explaining about 60% of the variation, which is pretty high. So we would be expecting that if the sativa in red and the indica and the hybrid explain the variation, perhaps we would see something like this, right? Where the sativas are in a corner with a particular chemotype, and the indicas are in another corner with a particular chemotype, and then the hybrids are in between. However, that's not what we see. What we see is that the sativas and the indicas and the hybrids do not explain the chemotypic variation. Again, this is a dimensionality reduction. So we're taking all of the variation in chemotype and we're showing it in two different dimensions, PC1 and PC2 in this case. But we see that it doesn't matter if you're an indica or a sativa or a hybrid, according to the industry, that doesn't say anything about your chemotype. However, if we partition this same graph by the most common terpene, we see a different thing. And we can see different clusters. We can see that in this corner, we have individuals that have high beta-myrcene, right? This is the most prominent terpene. And we can see that over here, we have the individuals that are high in terpinolene. And then when we take this same graph and we partition it using another mathematical algorithm, we can see that it clusters in three different clusters. One cluster that is high in caryophyllene and limonene, here in pink. Another cluster here in blue, high in myrcene and pinene, and another cluster here high in terpinolene and myrcene. So it seems that terpenes are better at clustering cannabis strains than the industry names. So again, when they tell you that sativas have a particular smell associated with a particular height, with a particular leaf shape, and indicas have another smell with a particular other height and another leaf shape. Be very, very skeptical. So to conclude this part of my talk, there are many genes related to terpene production. Are all these genes interacting to produce a terpene phenotype? Again, that question remains. And terpenes seem to be better at grouping individuals than industry names. And to conclude my talk overall, there is a lot of phenotypic and genomic diversity in cannabis. Perhaps we can breed for strains that have not yet been developed. Cannabinoids and terpenes show a mild correlation. It seems that there could be genes that are affecting both. Cannabinoids are highly correlated between them. And there's multiple genes related to cannabinoid and terpene production. These genes are scattered throughout the genome. Terpenes are better at grouping strains than industry names. And many important traits by the cannabis industry are not related to each other. So again, when they tell you about indica and sativa, be skeptical. And again, I want to thank all of my collaborators from academia and the industry. And I want to give a shout out today on 420 to all of my friends. Most of these are in Colorado, so I want to say hi to all of you. And my new friends here in New York State. And with that, visit our website, Cornell Cooperative Extension. And this is my contact information, and I am happy to take any questions. Thank you.
Outstanding, Daniela. Thank you so much for that wonderful talk.
Yeah, that was fantastic.
Thank you.
First question. Do the neutral forms of cannabinoids appear in the plant in low concentrations, like during storage or drying, or in a warm environment, or heating from overhead lights or the sun?
Yes, they do. Yes, definitely. Yeah, we do see that the neutral forms accumulate in very low quantities. You do have to heat them up for them to accumulate in higher quantities, but yes, you do see a little bit. Yeah.
Awesome. Next question. What is your opinion on the use of feminized seeds in the cultivation and breeding of cannabis? Has the breeding done by the legacy market contaminated the cannabis genome through breeding with feminized seeds?
That is an interesting question. So I don't know, contaminated, I wouldn't say that. You know, when you're breeding, you're choosing for a particular trait. So I wouldn't say that it has contaminated. Now, so feminized seeds, so what are feminized seeds? So something that I didn't talk about in cannabis is that when you stress the plant, so that means that you could apply particular hormones or that you can change light regimes, there are certain genes that are turned on, and you can have a female and you can make that female produce pollen. So then you have a female that has 2 X chromosomes, and that female produces pollen, and you take that pollen from that female and self-fertilize her, or fertilize another female friend. So then the seeds that are produced do not have a Y chromosome, right? So you're assuring that those seeds do not have a Y chromosome. They only have X chromosomes. That's why they're called feminized seeds. Now, several things. You don't know whether indeed those individuals, those seeds, whether they're not going to have the possibility of producing pollen if you stress them out or if you apply hormones. Perhaps they do. And in fact, many of the feminized seeds, there is a low percentage of individuals that do produce pollen. So that regarding feminized seeds, I think that feminized seeds are great. If they work for you, why not? Now, the legacy growers, the legacy growers have a legacy, and they have the word legacy attached to them for a reason. I think that in general, the breeding in cannabis has not been very very stringent for many reasons, right? Because cannabis has been illegal for so long that people have been breeding in their closets and their basement, literally, right? So it's really hard when you don't have enough space in your closet or in your basement to do good breeding, to breed for multiple individuals. You don't have space. You cannot, grow 300 individuals, 500 individuals that you need when you're doing breeding. So so in that sense, I do think that legacy growers have worked with the things that they have in hand, and that they have done really good things with the possibilities that they have available, I think.
Awesome. Next question. Were you able to, or do you believe it's possible to accurately determine cannabinoid levels in a plant, marijuana or hemp, using only leaves?
Cannabinoid levels, I don't know. I think that you need the flower, but what I do know is that the ratios of cannabinoids, if you do it even when the plant is little, the ratios do not change that much through time. And I'm not sure, I know that the leaves, and especially sugar leaves, those have a lot of trichomes, so they are going to show cannabinoids, but I don't know if they're going to show as much cannabinoids as the flower. Perhaps the ratios are going to be the same, but the amounts are going to differ.
Great. Another question. Can you speak about variation in flowering time? And would you speculate that shorter flowering times may have some relationship to C. ruderalis?
Flowering time. Oh, that's Larry Smart's baby. So can I speak about flowering time? We know that there is variation in flowering time, right? We know that there is, that there is variation in flowering time. And we know that depending on where you are or how many plants you want to grow in a given time, you may want to play with flowering time. If you want to grow several, if you want to grow in one season, you want to grow multiple plants, you want to play with flowering time. Ruderalis. It seems like ruderalis is a thing. We still don't know, like up north in Siberia, it seems that it is a thing. Again, we don't know whether there's wild cannabis. We don't know whether the cannabis that it's found out is feral, it escaped, or if it's indeed wild. We still do not know whether they're wild populations. And it seems that the ruderalis is a thing indeed. You see these plants that are shorter and that are out of flower. And so they do not obey the light cues, right? So usually cannabis starts flowering when it senses that the light is diminishing during the fall months. But yeah, ruderalis, it seems that there's some plant that there is out of flowering plants. There are out of flowering plants. Now, whether or not those genes are coming from ruderalis or not, whether those genes indeed are associated to these plants, I think that that's a different question. But we could answer that question if we have sequencing of all of these ruderalis plants, and if we have good phenotyping data, we could answer those questions.
Great. Another question, many questions. If you color the genetic PCA by terpene chemotype, is there any clustering there?
If I color the terpene PCA by genetic phenotype, oh.
Yeah, the genetic PCA by terpene variation, yeah.
So what was that genetic PCA? Are they talking about are they talking about the split tree graph? Or because I don't think that I showed a genomic PCA, but okay, so I don't know, I didn't do that, but so far I am showing here different data sets, right? In this talk, I show different data sets. Now, in an ideal world, we would have all phenotypes from each of the data sets and all genomics from the other data sets. For example, with the Leafly data set, we have a bunch of, tens of thousands of chemotypes that we had available thanks to Leafly, but we did not have the genotype of those individuals, right? Those were actually 6 different labs across the US, a lab in Florida, another in Michigan, another in California and Washington, that people go to those labs to measure their cannabinoids and their terpenes. So we do not have pairing genetics data sets for them.
That would be a really interesting question, though, right? Yeah.
Mm-hmm. And expensive as well.
Oh, of course, everything's expensive.
Yeah.
Another question, kind of unrelated. So you mentioned where the labs were that generated that cannabinoid and terpene data. Do you know where the dispensaries were? Were they in the same states as where the labs were?
I don't know. I know that the Leafly people do know. I was really excited to be part of that analysis because it's one of the biggest terpene cannabinoid chemotypes data sets that we've analyzed, and it was one of the best collaborations that I've had so far. And the 4 of us would meet on Fridays every 2 weeks, and it was a great collaboration. I know that there's 6 labs, independent labs in 6 different places in the US. I was positively surprised by the data set because the labs were very consistent with each other. The measurements were very consistent with each other, so I was positively surprised. I don't know if Leafly is going to make available the data, or if the labs made available to Leafly the data from the dispensaries or from the people, which I actually think that it's not important, right? I mean, what's important is that, one, the labs were very consistent, and congratulations, cannabis industry, that despite still being federally illegal and still having so much restrictions, the labs are doing great job at measuring these chemotypes. So first, and second, I think that gives us, it gives us peace of mind that there are labs that you can take your cannabis and they're gonna do a good job at measuring it. So I think that that's independent of who the dispensary was, or whether that was, I don't know, Purple Kush or Blue Dream. That doesn't matter. We have the data of the cannabinoids and terpenes that these strains had, and it's tens of thousands of strains. So we were able to measure and do a bunch of different statistics, and it was a very thorough statistic analysis that we did with that.
Awesome. Another question. Is there evidence that terpenes or groups of terpenes are responsible for the sativa hybrid indica effects that are persisting in industry marketing?
No, and that's what I just showed, right? So let me share my screen again. So this is the data that I just showed, that like the industry says that so the industry says that indica sativa hybrid, you have these particular characteristics which are not correct, right? We don't see that with the genome. We see that indica sativa hybrid, they are not related in any sort of genotype, right? And we don't see it with the phenotype either, whether we look at phenotypes like leaf shape or height or branching or nodes, or males or females, or terpenes or cannabinoids. So that indica sativa hybrid is, I don't know what the basis is for that indica sativa hybrid. Like, it's kind of like saying, like kind of relating, like, okay, if you are blue-eyed, you're always gonna be blonde. Like, that has nothing to do with each other. Or if you are tall, you're always gonna have brown eyes. That has nothing to do with each other. Those traits are broken by recombination. So if we see this graph, right, we would be expecting, I mean, this is my made graph, like it's not as pretty as what we actually really did, but if we would be expecting, if the indica sativa hybrid if it were true, we would be expecting that the indicas would cluster with each other and the sativas with each other, and maybe the hybrids are somewhere in between. But that's not what we see. We see that the names are not related whatsoever with the type of compounds, but we do see that we can cluster these strains based on the most common terpene or groups of terpenes, right? Here in this graph, when we do this clustering analysis, we do see this clear partition, and this clear partition is, we named it cluster 1, cluster 2, and cluster 3, but you can call it, I don't know, Mary, Jim, and John, right? These 3 clusters do have particular terpenes, which are the most abundant terpenes. So terpenes are better at clustering than the indica sativa hybrid. I don't know if that was clear.
Awesome. Another question. What do you think are the technical or computational bottlenecks for completing the whole cannabis genome?
The technical or computational, I don't think that there are any technical or computational bottlenecks. I think that the bottlenecks are money, because you require someone that knows a lot of bioinformatics, and more money to sequence a really good Y chromosome with long reads, you know, like a PacBio type of thing, long reads, and very high coverage. So I would sequence a bunch of males with very long reads, with very high coverage in both PacBio and corrected with Illumina. So that's some chunk of money, and then you would need a lot of people, good people that have really good bioinformatics skills, that have a lot of time to dedicate to this, to do that. I think that one of the reasons why the Y chromosome is not finished, it's because it's hard, it's huge, and it has a bunch of repetitive content. So it's really hard, you know, like you have blah, 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 blah. It's really hard to put it together. It's really hard to build that puzzle. So I think that it needs a lot of time, a lot of money to do it.
Okay, I think we'll do one more question, and this will kind of bring us right back to where we started here. How do you think that we preserve the diversity in the cannabis genome so that we don't lose possible therapeutic effects, and I'll add, you know, other diverse characteristics that may be present in the plant but have been overlooked in the past?
Preserve the cannabis genome. Well, I think that that's what Zach and Tony are trying to do, right? I think that that's why the USDA and that repository is so important, because that's the entire point, right? Preserving all of these different strains, all of these different varieties, in order to have a good library that represents the variation that's out there. And I remember that when Zach and I met, that was one of his questions, which I pondered and I discussed with multiple people, like, how do we know that we are indeed having a good representation of what's out there? And if indeed those preliminary results are right, that cannabis has 2 or 3 times as much variations as humans at least, how many individuals do we need to have in that repository that are representative of what's out there? And what I recommend to people is, you know, keep seeds. And then if you see a feral little guy and you see a ditchweed, do not ditch it. You know, it may have really good characteristics. And yeah, so I think, or at least, you know, try to give it to Zach somehow. You know, I don't know what are the laws and the regulations with the USDA, but that is why Zach's and Tony's job is so crucial and so important.
So just very, very briefly, I just want to speak on everyone's behalf, because I think it's true. That was a fascinating, mind-blowing, informative talk, Daniela, and you are a treasure, and thank you for your time. That was one of the highlights of the entire seminar series. So thank you again. And to the audience, we are very interested in hemp genetic diversity and conserving it for all of the reasons that Dr. Vergara illuminated. Please reach out to Tony and I if you think you may hold some genetic diversity that we can conserve in the long term. I just was curious, your vision of 5-10 years for hemp genetics, 20 years, what could that look like?
Mine?
Yes.
Oh my God, that is such a good question. First, and such a good question on 420. Please have a happy 420, everyone. But you know, I've been thinking about the question now that I'm dealing. My job now requires me to work with people and with farmers, and I am gonna say something that may not be that popular among people, but I think that big companies and big corporations, which I'm not gonna name, I am not against them. I think that many of them have done really good science. However, I do think that there is a disconnect with what they propose and what the public gets. I think that people need to be educated about genomics, not because you're all gonna become genomicists and bioinformaticians, but because when you understand it, you make better choices and educated choices. I think that it's really important to know who is going to have this data. GMOs are an excellent choice, in my opinion. GMOs and vaccines are kind of like mind-blowing, like, you know, like a movie, you know, like I think about it and it's like, wow. And it's going to happen, but it's who is going to handle that data? Who is going to have access to that data? Is it going to be few stakeholders, which is what we've seen so far? Or is it going to be part of the public realm, right? Like, are we all going to have access to that? And I think that that is really important. And I think that cannabis right now is in that point, right? There's many companies that are looking, for example, flowering time genes. There are several companies that have their eyes on flowering time genes. Who is going to hold those patents and those technologies? Is it going to be 2-3 companies, or is it going to be all of us? So that's something that I hope that cannabis differs from other important crops. Maybe a romantic way to end the presentation.
So I think we'll leave it at that. Thank you so much, George and Tony, and everyone that's contributed to this. But once again, thanks again, Dr. Vergara.
Thank you. Thank you very much.
I think we could have kept you in here for another hour just answering questions.
Yeah.
Really? Were there? I'm looking at the Q&A. Where indica and sativa and hybrid diet or where the products came from. Oh, these are really good questions. As a follow-up, what is the thought of the difference? Oh, I know Matthew Indest was here. Oh, I know some people. So as a follow-up, so from Matthew Indest, what is thought to cause the difference in effect? I mean, I don't know the difference in effect because we have so many compounds interacting with each other. That entourage effect is a very, in my opinion, it's a really interesting hypothesis. And I think that you have different ratios of compounds and multiple of them. So if they're all interacting with each other, and besides, we don't know, you know, if the smoke that you, the joint that you smoke today, is it going to have the same effect as if you do it tomorrow, right? Or is it going to have the same effect on me as on you? We don't know that. So those are really important, interesting questions. Were the indica, sativa, or hybrid data provided by the dispensaries where the products came from? Yes, this data was provided to Leafly from the dispensaries or from the people. So yes, yeah, those were the names that the industry provided. Does the biosynthetic pathway help explain the correlation of terpenes and cannabinoids? Zach Spangler, I've seen Zach Spangler before. Yeah, I think so, right? Because they're using the same precursor molecule. So, and then cannabinoids and terpenes at some point, they come from the same biochemical pathway, right? Several steps above, they do come from the same biochemical pathway. So yes, I do think that they do explain the correlation between terpenes and cannabinoids at some point, but there are multiple genes that are interacting, right? So, okay, what other, can you measure the amount of cannabinoids within the hires for industrial hemp production? That would be really interesting to do, to measure the amounts of industrial hemp. However, because it is not required, then people do not do it, right? If you're growing it for fiber or for oil, you're just going to measure THC to understand whether it's compliant with the law. But do you really care about the other terpenes? However, I do think that that would really complement the scientific story. Angeli, are the terpene synthase genes linked with the THC or CBDA genes when you showed the PC analysis? So for that PC analysis, because we had so many high THC strains, we used those, right? Because they were from dispensaries, and people go to dispensaries for high THC mostly. Thomas, are there any studies and collections completed in progress on American feral hemp? No, but we can start one, I think, or SAC.
There was one that came out like 2 days ago on Nebraska feral hemp.
You're right. Yes, on Nebraska feral hemp. So yeah, there are. Do the number of cannabinoid genes affect the amount of cannabinoids a plant produces? So 8 equals X and 32 equals first X. We didn't find that. We did find that there were some positive correlations between the number of copies and some negative correlations. So we don't know. For THC, for example, we found a positive correlation, but for CBD, we found a negative correlation. So that's a good question. Do you consider doing a PC analysis among phenotypes of flower and its correlation to cannabinoids quality and quantity, since that genetic plays an important role in specific genes are expressed? Yes, but I don't know if a PC analysis would be the right way to do that. I mean, I love PC analysis because they're so versatile, but anyway, I think I answered many of these questions.
I was curious. Sorry, here we go. I was fascinated by your statistically significant correlation between terpenes and cannabinoids, and that could be an example of a pleiotropic or epistatic effect potentially. Do you think that could also be explained by agricultural conditions? Healthy plants produce more secondary metabolites in general?
That's a great question. That's a great question. I think that it could be. However, there are many terpenes that are produced when you are not in the best conditions, where you're being eaten, right? So in certain conditions, you may not want to you're producing those terpenes because you're actually stressed, like you're getting eaten by a worm. So you're producing those terpenes. So I do think that, like we know that light plays a crucial role in the production, at least of cannabinoids. So perhaps when you have really good light, you produce more cannabinoids and therefore more terpenes. We also know, again, I would like to know more about the genes related to trichomes, right? If you have bigger trichomes or more trichomes, are you also having more abundant cannabinoids and terpenes? But I do think that measuring those environmental effects and having a constant environment in order to do this, I think that would be crucial. That's a great question, I think.
Any work on a cannabis pangenome?
Sorry?
Do you know of any work on a cannabis pangenome?
Well, that's what people are suggesting, right? That's where we're going, right? Again, I think it's important for academic institutions to be involved in that and to try to have students be involved with data collections. But yeah, I think it's a great way to do. I think it's important to have these pangenomes. Yep.
Well, thank you for staying a little extra. And, I want to talk to you all day, Daniela, about this stuff, but we should probably let you go.
Okay. Thank you very much. I had a lot of fun. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.